to also produce the pluripotent stem cells. You know what I meant by embryonic stem cells from the blastocyst? We took the fibroblasts from the animal, we put the Yamanaka factors, the 4 transcription factors, and we induced adult cells, or actually the fetal fibroblasts, differentiate cells actually, to become embryonic stem cell-like cell, and that's called induced pluripotent stem cell. So we have 2 types of pluripotent stem cell line: embryonic stem cell line, induced pluripotent stem cell. Some of my students have done great job on this, and then these are all there in the public domain. So we have these 2 different cell lines. Both will be identical in their properties, but the method of derivations are different. And one is ESC, the other one is iPSC. So this is how these cells look. They form actually colonies, and you have these little colonies here. So each colony may have 50 to 100 cells, and they have a very nicely compacted colony. This is not compacted embryo, but these are embryonic stem cells or induced pluripotent cells, which are compacted, which has got nice distinct boundary around it. Usually each colony has got 50 to 100 cells. This is a single colony. This will have about 50 to 100 cells. Because these are from the transgenic mouse, so every cell will be fluorescing. So every colony you find here, these are multiple colonies of about 10 or 15 are showing here, is showing 4. There is a single one at different optical magnification, and they are how. And how do you know that these are ESL? Anybody can challenge, hey, we have something, some colonies. What if it is some other cell type? So there are a number of defining methods or criteria you have to go through rigorously to say that these are established embryonic stem cell line, or these are established pluripotent stem cell line, that is the induced pluripotent stem cell. We do about 10 or 12 tests to confirm this. Only then you can say that this is a pluripotent stem cell line. Otherwise, the journals won't accept. Other scientists will not believe you. So one is that this should be able to propagate endlessly. I can give it to every one of you, and then you should be able to do this. So which means you have established a cell line. But do they have the property of other things for the typical ES cell line? Remember, they have to form 3 embryonic germ layers, and they should form organisms. So we go through all this, and there are a lot of other molecular criteria, because that there are some stem cell proteins which these cells should express, and some very stem cell specific associated transcription factors. OCT4, octomorph4 transcription factor, which is nucleus localized, should be expressed by each one of these cells. And therefore, one colony here I'm showing OCT4, which is expressed in the nucleus. Why am I saying these are expressed in the present rather in the nucleus? And if you do a nuclear staining with the DNA binding dye, those are the blue cell, blue organelle or the nucleus. And this red color and the blue color gets co-localized, and you get a pink color, which means you can definitively say that the OCT4 is expressed and they are nuclear localized. You take another example of another protein which is expressed by the stem cell. This is what is known as stage-specific embryonic antigen 1. Don't worry about name. And it has got its own initials, SSEA1. And this is the plasma membrane, you know, the cell's outer plasma membrane. And that gets present only in the plasma membrane, and that's why you see in the plasma membrane. So those cells, if you look at for the DNA is stained by the DAPI fluorescence, SSEA by this red fluorescence, they don't merge like here. So you know very specifically that these are things. So anytime you take these cells and do this characterization, they should produce. This is what is known as immunofluorescence phenotyping or molecular phenotyping. You can also mix these cells with the wild type cells from the blastocyst. You remember blastocyst and the inner cell mass. If you mix these 2 cells, they are all having the same property. Inner cell mass of blastocyst and these cells have same property because they have originally come from there. They will mingle very well and form those embryos, and eventually the organism formed are called chimera. A chimera is a mix of two different genotypes. So it will have properties of this genotype and the host blastocyst cells phenotype. So we formed chimeras, and these are typical example of chimeric blastocyst formed. And if you transfer this, you get the chimeric mouse. So this is the most important evidence that the ESL that you produce is actually a totipotent cell. It has formed the organism, and these are not totipotent, and therefore you are putting them into blastocysts. You remember, are making them to form blastocysts. So that's why this method of chimerism is important to demonstrate the pluripotent stem cells that you derived are formed. The other method is you can inject into the animal, and that tissue, sorry, I don't have that slide here, something is missing here. If you take the tissue and make an histology of whatever those cells disorganizedly formed in an animal, you will actually see ectodermal tissue, mesodermal tissue, and endodermal tissue. So if you give the tissue sections to histopathologists, hey, you have a mixture of these cells, man. This is not liver, this is not lung, this is not heart, this is not brain. You have some of the cells, some of that cells, it becomes. So what it does is, because it is not in the uterus when I transplant it to subcutaneously, it's forming underneath the skin. It is trying to form embryo, but in a very disorganized manner because it doesnn't have the uterine maternal environment. And therefore, in a disorganized way, embryogenesis has taken place, forming ectoderm, mesoderm, endoderm, and ecto-meso-endo-derived cell types. So the histology will show all the cell types. There's a beautiful slide we had, which is published there in that paper, that will demonstrate. So that is one. What is the other method? The other method is actually to make these stem cells in vitro to form ectodermal cells, endodermal cells, and mesodermal. Experimentally, you can make them to form ecto, meso, endodermal cells by some kind of an experimental induction of differentiation. So this differentiation biology is extremely important, and scientists, stem cell biology, stem cell scientists are very importantly working on how to make ecto-derived neuronal cells, endo-derived pancreatic cells, meso-derived cardiac cells, or muscle cell, skeletal muscle cells, or smooth muscle cell, so on and so forth. And because these cells are required for transplantation for diseases which are specific to organs. And therefore, you should be able to demonstrate the in vitro differentiation of pluripotent stem cells, whether it is ES cells or iPS cells. So this is a holistic picture of defining criteria when you have established ES cells or iPS cells. So we have these two lines which we have established and we have produced. So I will tell you something about the differentiation, and we have done this with our own transgenic animals, GS2 ES cells, N9 iPS cells. We have also done wild type, which means it's not expressing fluorescence. So wild type cells, non-fluorescing cell type. We did this both in the mouse. We also did this in the human. We did a lot of work on those, so I will not go into the detail. I'll only demonstrate 2 slides how to show or bring in differentiation. You can make these stem cells to undergo differentiation. If you want to make them to undergo differentiation, one of the methods is to form what are known as embryoid bodies. These are aggregated cells, form like pebbles, and they form, you can make hundreds and thousands of them. And how you make this? You disperse the ES cells, some 20 or 30 of cells, or 50 cells, or 100 cells. So you put 100 or 200 or 400 cells into small drops, and we hang the drops here in a lid of a petri dish. I mean, there's a method. So you take the lid, put these drops, what you see here, and then invert them and put it into the main dish. And therefore, they're hanging as if these are little leaking drops of the rainwater in your ceiling, roof ceiling. And so they are hanging with those 400 cells or 500. They multiply, but the space is limited. When the space is limited, like that morilla formation, embryo compacting, they try to compact and form what are known as a very spherical embryoid bodies, and those are called embryonic bodies or EBs. That way you can produce thousands of them. Depending on how many microdrops you have made, you can make 20 microdrops of each. You can make 5,000, 100, 2,000, whatever number, you'll get that many. So this actually has got a mixture of ES cells. It will also have some ectodermal cells, mesodermal cells, endodermal cells, differentiated, but they cannot go any further. If you want them to go further, you take each one of these embryonic body, put it separately in a dish, give them space. Put them at the center of the well. They will attach and they will proliferate. They proliferate like crazy. Like you have a central embryonic body which is attached and they just go all over the place. It fill up the entire petri dish. So they differentiate. They form ectodermal cell, mesodermal cell, endodermal spontaneously. So here is one embryonic body attached, proliferated. It takes about 5 to 8 days. And you actually see some cardiomyocytes formed already by day 9 or 11 of attachment. So you will see beating cardiomyocyte, beating patches. You will have about 10 or 11 of them in this outgrowth. And I'll show you another slide of only a single cardiomyocyte beating patches. So these are cardiomyocytes, functional cardiomyocytes, because they're beating. You can trypsinize this and get pure population of cardiomyocytes. So now you have a method from ES cells or iPS cells to produce functional cardiomyocytes. You can also take precursors of that one step before, that's called progenitor cells. So you have cardiac progenitor cells, you have cardiomyocytes. Cardiomyocytes are differentiated functional cells. Cardiac progenitor cells are these precursor cells, which will eventually go and differentiate. So you can take cardiac progenitor cells. The question is, how do you make them in more numbers, millions of cells? You need at least, let's say, 2 to 3 million cells of cardiac progenitors or cardiomyocytes per kg body weight. If I am 65 or 70 kgs, I need 140 million cells to inject. Where do you get this cell? And you know how tiny these cells were. That's why a scale-up biotechnology aspect of stem cell research come. How do you make more ES cells, billions and billions of ES cells, and out of that, how do you make millions and billions of cardiac progenitors or cardiomyocytes? This is the big deal in stem cell differentiation biology. And that's where stem cell scientists, stem cell technologists work very hard to figure out what is the recipe to get this to undergo differentiation, and so on and so forth. How to make them in large numbers and how to preserve them, cryopreservation, so that it can be used on a later date, so on and so forth. This is about cardiomyocytes. So this is a purified population of cardiomyocytes that you actually see. Now, this is not a cardiomyocyte. The other one I showed is not a cardiomyocyte. It's something spontaneously that forms something like the smooth muscle. Smooth muscles are there. For example, blood vessels, your intestine, all these have got smooth muscles which don't listen to you. Skeletal muscles are muscles in our bone, attached to bones or skeletal muscle. You decide and you contract. I am saying my fist should be closed. I'm able to do. I can't stop my smooth muscles of my body. Either it is respiratory tract or intestine. It automatically keep doing it. Blood vessel will just contract like this to make the blood flow. They are the smooth muscle and these are the smooth muscles. And it has got a different contractility pattern. And this is skeletal muscle. This skeletal muscle will have kind of a twitching type. You see that these are twitching. This is different from cardiomyocyte contractility, smooth muscle contractility. So these are my tubes. Now, somebody may ask, okay, fine, you're able to produce mesodermally derived musculature, but how do you make, let's say, neural cell types? You have different neural cell types. You have supporting neural cell types, glial cells, astrocytes, oligodendrocytes. You have neurons, different types of neurons are there. GABAergic producing neuron, cholinergic producing neuron, dopaminergic producing neuron. Because these have very specific target functions in our brain and a peripheral nervous system. And how do you get them? And so from stem cells, we were able to do this. I don't want to go into detail, but there's a particular medium called a QASR medium. So unfortunately, these are commercially available medium. It turned out one of my students, Disha Verma, did this experiment. When she did this embryonic body formation in that medium with some specialized conditions, they predominantly produced neural cell types. So it has given a mixture of neural cell types, which is astrocytes, oligodendrocytes, and neuronal cell types. And there are umpteen number of neuronal cell types, and these have to be typed. And so she spent a good 5-6 years to figure out how the neuronal cell types are formed, what proportion these are formed, and when they are formed. And there's a whole biology she did for about half a dozen years. And she did this. And this is the beauty of stem cells. So you can do this from the ES cells, you can do it from the iPS cells. So if one is interested to produce neural cell types of particular type, you can do this. Now, I'm going to tell another big challenge. And the big challenge, like I was mentioning, is how to make stem cells in large number is one big challenge. And the other big challenge is how do you make them to differentiate? And I'm giving a schematic example of how one can make it to differentiate to cardiac progenitor cells derived from pluripotent stem cells, whether it is ES cells or iPS cells. The challenges are culturing them and differentiating them. The second challenge is once they are differentiated to what? Cardiac progenitors, cardiomyocyte, neural progenitors, neuronal cells, or pancreatic progenitors or pancreatic beta cells. So if we want only the cardiac lineage cell types, then you have to come up with step 1 and 2 is only that, and step 3 to enrich them and characterize them. And lastly, expand them in large number. These are the big challenges in differentiation biology in terms of differentiation, in terms of identifying them and enriching them, and after enrichment in terms of expanding them to a sizable proportion which can be used for transplantation. I'm showing here the mouse, and replace this mouse and put a human being and look at all these as human cells. You can do this. And that's the whole kind of biology which is extremely important in stem cell research or stem cell technology. We have a headway. I will not go into the detail except maybe 1 or 2 slides I'll show. We have a method to culture them and make them in more numbers. We have a method to enrich them. We have a method to scale them up and then produce in large numbers. And we're just there to do transplantation. If you want to do transplantation, you don't have animals which are naturally susceptible to develop, let's say, heart attack. So sometimes you need to experimentally induce a heart attack type of thing, or what do we call as experimental induction of cardiac ischemia. So that's simulating animals which may have an heart attack if it is not cured. So if one can do that, if you transplant these cells, you will be able to show that it is experimentally proving to help the animal in terms of not getting ischemia and not getting heart attack by doing a lot of cardiovascular physiology on the animal and so on and so forth. So you can try to demonstrate these in non-human species, and then you can demonstrate this perhaps by clinical trial in the human. If you use human cells and if you use fit enough human cells, they are called clinical grade human cell. Then the entire process of doing this should be in an absolutely sterile environment called the GMP grade environment or GMP facility, in which everything has to be done there. So that's a different ballgame. Maybe tomorrow I'll tell a little bit of that. So the whole process of these activities will really make the stem cell biologists and stem cell technologists to be very, very busy at each one of these steps. And each one is a big challenge, and it requires a lot of expertise and technology to work. One method of enrichment is that some of these cells that I'm showing may have a plasma membrane protein which is very specifically associated, if not exclusively, associated to the cardiac progenitor cell. And if there is a method to enrich those cells, then you can specifically enrich those cells. And that's what is known as magnetic assisted cell sorter. I will not go into the detail. So basically what it means is that you can have an antibody to the protein of the plasma membrane, a plasma membrane protein, and you have developed an antibody, and those antibody can be tagged with a magnetized particle, which will get attracted under the influence of magnetic field. So in that way, you can just produce those and enrich them. So there's a method of enrichment and there is a method of expansion of the same cells. And you can, I'm showing here from 0.5 million to 1.2 to 3.6 to 34.6 million. That way you can just expand, and there is a method to do this. So this is pre-enrichment proportion and this is post-enrichment proportion. I just want to make sure that you people are hearing. Can somebody say hello? Can you all hear so far? Okay, good. This is not the mouse but the human. So you see that these are human. These are human induced pluripotent stem cells, which are induced to undergo differentiation to functional cardiomyocytes, human cardiomyocytes. You see them. And once they are differentiated, they peel off and they float. So you can just cook these ones, trypsinize, and keep them ready for transplantation, let's say, into the human, so long as this is a clinical grade, which is fit enough for transplantation in the human, the cell quality. But these are laboratory grown cells, so we cannot use this for human transplantation, but this is good for understanding differentiation in vitro in our research laboratories. So this is the iPSC of humans differentiated to cardiomyocytes. So this is another way where, for example, the previous one, what I showed here, is in a petri dish. It's called 2D cultures. Suppose if I make these cells on a small filter paper, it's not the regular filter paper that you or the lab may have. It's a very specialized biocompatible filter paper using some biocompatible organic molecules. And that filter paper, you can make the cells to form. So it's like a scaffold, paper scaffold on which these cells are grown. iPS cells are grown to form cardiomyocytes. So what is the advantage of doing that on a paper scaffold like this here? You can simply take this paper, small piece of paper of 2 millimeter by 2 millimeter, put it on the infarcted heart region and stitch it up. So it's like a small paper with cardiomyocytes stitching onto the cardiac tissue where there is an injury, tissue injury. So these cells now will go home in the infarcted region and form functional cardiomyocytes in the defective place of the heart. And therefore, the ischemia can be overcome. So these are PCR gelatin scaffolds paper on which human iPSC-derived cardiomyocytes are formed. And this is the translation, which is very, very critical. And this can be doable, and it is done being done with the human cells. It is being transplanted successfully in small laboratory animals, experimental, and also large mammals like domestic animal like pigs. And therefore, these approaches will become successful soon, rather, I would say, and then can be potentially be used in the human. So these are heavy amount of human research and preclinical research that one needs to do to reach this stage for potential transplantation of stem cell derived cells into the human. Okay, so this one example of what I just now showed, what we produced here. And these are those little scaffold, small little paper. This is 2D culture, this is 3D culture. And some British scientists produced similar types, what I'm showing on the top, and this is what we show. So these are the potential benefits of working on stem cells. And I'm going to stop my presentation for today here, and then probably tell more on the application point of view on the stem cell biology and technology tomorrow. So I will stop here, and I will be happy to take some questions. I'll share this.
Thank you very much, sir. The session is now open. The session is now open for interaction. Any doubts?
Good afternoon, sir.
Yes.
Sir, how can we differentiate embryonic carcinoma cells and cancerous cells, sir?
Embryonic carcinoma cells have got similar properties as embryonic stem cells, whether it is ESC or iPSC. So the differentiation path will be similar to what we do for the PSC cells. But embryonic carcinoma cells, you said cancer stem cells, right? Yeah, the cancer stem cells have the property of only one cell type. It is a unipotent cell type. So if it is a liver cancer stem cell, it is capable of producing only liver cancer cell types. It is different from the embryonic carcinoma cells. You understand the difference? Cancer stem cell will form only the same type of cell. If it is heart cancer, it will form only heart cell. Heart cancer is very rare, because by the time it forms, the patient will die anyway. Liver, kidney, mammary tissue, prostate, what have you, cervix, all these form stem cells which will in itself form only that type of cancer cell types and not any other type. So the difference is there.
Sir, another question. Is the same, you said about some factors involving the differentiation of iPSCs, like output, SOX2 like that. Is the same factors involved in the initial development of the field cells?
If you make that statement without a question tag, the answer is right.
Okay.
You understand what I'm saying? What you said is the answer. Yes.
Another question. You said about diagnosis hatching. Is there any particular area of hatching takes place?
Good question. You have done a lot of work, and if you have a blastocyst and you have inner cell mass in one side, in this on my hand here, and it is not here, this is called embryonic side. This is called the abembryonic side. Okay, and hatching in some animals occur in this side, abembryonic side, not embryonic side. It is believed that the human also may be an abembryonic side, opposite to embryonic pole. This is embryonic pole, this is abembryonic pole. So it's very likely it is happening here. Abembryonic pole or mural side.
So is there any surface markers to differentiate between the different types of stem cells? Like the hematopoietic stem cells or mesenchymal stem cells?
Yes. Hematopoietic stem cells can be differentiated by the expression of CD34. It is the cardinal surface marker. And those cells which do not express CD34 but express 70 and 90, CD70, CD90 will be mesenchymal stem cells. Hematopoietic stem cells produce CD34. So there are a number of cluster markers. So you can differentiate, you can enrich also. Yeah.
Any other questions?
Sir, good afternoon, sir.
Yeah.
Sir, Sir, can you please explain us how the normal animal and identical twins are formed?
If I can repeat your question, can you please explain? Identical twins is where the oocyte fertilized by single sperm and form two cells. For some reason, the two cells separated off, and independently, I demonstrated in the petri dish. You can take it out and culture them. So if you do that from the monozygotically fertilized oocyte and have those daughter cells forming differently, two years going parallelly as 2 cell, 4 cell, so on and so forth, they are identical twins. Did I make it clear? They are clones, identical clones.
Okay.
So identical twins are clones actually.
Okay. Sir, one more question, sir. Sir,
Go ahead.
you know that in some state only the division takes place and not the differentiation will not take place, sir. Only the celestial divide and the differentiation will not take place in the
Why is that? Maybe because the auditorium. Can somebody repeat the question? I can't hear. Can you say slowly? Can you repeat the question?
Yes, sir. So at which stage the cell differentiation takes place?
I mentioned in one of the earlier slides, after fertilization, you have the zygote, and zygote forms two cells. These two cells become 4 cells, 8 cells. Until then, there is no differentiation. But when there is a crowding of cells within the zona pellucida, you know, the outer case, there's a lot of crowding inside. When there is a crowding and the cells are tossling with each other, that is the initiation of differentiation. And that tossling happens with the cells which are outer to the ball of cells. So those cells begin in differentiation to trophectoderm or epithelial-like trophectoderm. So the first sign of differentiation is forming morula, compaction, and the cells that are around on the surface. That's the first time of embryonic differentiation. We all went through that. I told you, 280 days before your date of birth.
Okay, sir. Thank you, sir.
Anything more?
Thank you, sir.
Yeah.
Sir, why is it difficult to actually cultivate neuronal stem cells?
Again, I got the letter half. Different from stem cells, you said. What the first stage?
Why is it so difficult to cultivate neuronal cells from stem cells?
Yeah, good question, because the neural cell, actually you can achieve it, and we have done it, and like I said, we have a methodology to do that. But these are very sophisticated cells. The morphology, cell biology is very different for neural cells. For example, neurons of different types in different parts of the CNS have a different shape and size and properties. And their dendritic structure and axonal extensions are different. And therefore, it's a very specialized differentiation process. Getting them to happen in vitro is very challenging, particularly to a particular neuronal cell type. For example, we want dopaminergic neurons for Parkinson's disease. Parkinson's disease is like, like people who have age onset, kind of a fits type thing. And there's a lot of disorientation. And the dopaminergic neurons, which is in the brain, substantia nigra region, is the one which controls. And you can get dopaminergic neurons in vitro. But there are other neurons which are rather difficult. particularly less abundant neurons is more difficult because they form in less numbers. Whereas GABAergic neuron and glutaminergic neurons are very predominant, 50, 60%, 40%. They are there, easy to form. So the low proportion, low abundant, or rather least abundant ones are more difficult. Mother Nature does, but we are still trying to understand because their in situ niche, the kind of biological niche during fetal development is different from what you give in the petri dish. We don't know what else they require, what they don't require. That's where the differentiation biology research is important.
Thank you, sir.
Thank you, sir. Next, I would like to call my classmate Aisha from 3rd Ph.D. Biotechnology to end session 2 with board of things.
We have to show present here gratitude. Always have an attitude of gratitude. Yes, it's my privilege and I'm really glad to propose. What are thanks for this special occasion? On this exemplary day, I would like to thank our brilliant and outstanding professor, Dr. P. Seshagiri sir. It is immense pleasure to this speech and great vision about central biology and technology principles. Really thank you, sir, for taking time out of your busy schedule to interact with us. I think that we are really blessed to have you here. Your sparkling words made this day very great and beautiful words for my head. So we have from the bottom of the heart. Thank you, sir. And once again, thank you for all the presence, sir. Thank you.
Oh, thank you. Okay. Bye for now. Bye.
Before moving on,